Let's discuss on the topic of the regulations of the gene expressions in the eukaryotes. And these are the introductions. What happened in gene expression in the eukaryotic cell, it is mainly regulated by the repressor as well as by the transcriptional activators. Like the prokaryotic counterparts, the eukaryotic repressors will come and bind to the specific DNA sequences and then it will inhibit the transcription. Other repressor will come and compete with the activators for binding to the specific regulatory sequences. And the gene expression in the eukaryotes, it cannot be same as that in the prokaryotes because of the genome that it is larger than the prokaryotes, and genes are mainly altered by the gene amplification, gene rearrangement, gene loss and the metallizations of the DNA. So these are the stages of the eukaryotic gene expression: the chromatin structure, transcription, processing and export, mRNA stability, translation and the protein processing. So let's discuss about all the stages. The chromatin accessibility. It is the structure of the chromatin, the DNA and its organizing protein. It can be regulated, more open or relaxed. The chromatin make a gene more available for the transcription. And the transcription is a key regulatory point for many genes, and set of the transcription factor protein, it will come and bind to the specific DNA sequences in or near a gene, and it will start to promote or repress its transcription into an RNA. RNA processing, what happened here, the splicing and capping and additions of the poly-A tailing will be taken place to an RNA molecule. It can be regulated and so that it can exit from the nucleus, and different mRNA may be made from the same pre-mRNA by alternative splicing. And mRNA stability, what happened here, the lifetime of an mRNA molecule in the cytosol, it will start to affect and it may be started to affect and it may be quickly degraded. And coming to the translations of an mRNA, it may be increased or it may be inhibited by the regulators. For instance, what happened, the mRNA, sometimes it may block the translations of the target mRNA rather than causing them to be chopped up. Protein activity, the protein actually it can undergo a variety of the modifications such as being chopped up or tagged with a chemical growth. These modifications can be regulated and may affect the activity or the behavior of the protein. Although all the stages of the gene expression can be regulated, the main control point for many genes is transcription. And later the stages of the regulations often refine the gene expression pattern are rough out during the transcription. So here we can see the DNA will be actually converted to the primary transcript. It will be, and here the transcriptional control will be taken place, and we can see here this yellow part. This yellow part, they are the introns. This intron will be removed out. After removal, what happened? The 5' capping polyadenylizations will be taken place. This here the 5', here the polyadenylizations will be taken place. After the RNA will be getting converted, it is like taken place by the RNA processing control. So after that, what happened? This mRNA will be converted to the RNA, and then what happened? This RNA actually will started to it will transfer to the cytoplasma from the nucleus to the cytosol. After converting, what happened here? We can see the protein that are the amino acid. These color blocks are the amino acid. So these are taken place by the translation control. So what happened? These amino acid are like folding will be taking place. After the folding will taking place, what happened? The modification will take place. So in this modified protein, they are known as the post-translation control. So we can see here like a total like five. It is a five control. That is a transcription control, RNA processing control, RNA transport control, and a translation control, and a post-translation control. And there are also environmental control. So we can see here all the stages of the gene expression. They can be regulated. The main control point for many genes is a transcription. So we will discuss here the genomic level, transcription level, post-transcription level, and the post-translational level. So come here in the genomic level, so we can see here the heterochromatin are tightly packed, and even the euchromatin. The euchromatin is a loosely packed form of the DNA in the chromosomes, while what happened while the heterochromatin, it is a tightly packed form of the DNA in the chromosomes. So we can see here it is tightly packed. The euchromatin actually it is lightly stained and but the stain dark during the mitosis, whereas heterochromatin it is stained darker during the interphase. Coming to the transcription level. So coming to the transcription level, what happened? The variations is in the rate of the transcriptions often it started to regulate the gene expression and the interactions between the RNA polymerase II and the basic transcription factors, and it started to lead to the formation of the transcription initiation complex, and it will influence the rate of the transcription. Other transcription factors it will started to change the rate of the transcription initiation by binding to the promoter sequences, and the rate of this transcription what happened started to influence by the enhancer and then silencer. So here we can see the promoter site where the RNA polymerase will bind, and the promoters are two types are there: the basal promoter, that is the Tata box, and the upstream control element, that is the GC box, CAT box, and the SP1 box. So here we can see these are the different boxes. So these are the eukaryotic promoter. So in the eukaryotic promoter, what happened? There are two parts. That is the core promoter and the basal promoter, and another one is the upstream promoter element. So here we can see the promoter. And the core promoter actually it is constituted by the Tata box and the transcriptional start site, that is a TSS. And the initiation complex will come and bind to the core promoter, and the upstream element will be responsible for the regulations of the transcription. So here we can see the enhancer. This enhancer in the some eukaryotic gene, there are the regions that will help to increase or enhances the transcription, and these regions actually they are called the enhancer and are not necessarily close to the gene they enhance. The transcription, the transcriptional factors will produce the constitutively, but except these three are the some transcriptional activators which will come and bind to the enhancer site, and these transcriptional activators are induced protein that is synthesized only in the response to the specific signal, which on binding with the DNA forms the loop back on itself and then started to interact with the TF near the promoter, and the interactions between the enhancer site and then in initiation site it is usually necessary for the transcription above a basal level. Coming to the silencer, what happened in the silencer? It is a DNA sequence that capable of binding the transcription regulation factor that is called the repressor. Like an enhancer, a silencer also functions irrespective of its positions that many thousand base pair will be aware, and the orientations relative to the gene whose expression will be controlled. Here we can see the activator and the repressor. So what happened? The some transcription factor activate the transcription. For instance, they may have the general transcription factor and or RNA polymerase will bind to the promoter as it is shown in the diagram below. So this activator will have the general transcription factor and the RNA polymerase to assemble, and the transcription here will be taking place. And a repressor, what happened? Other transcription factor repress the transcription. This repression can work in a variety of ways. As one example, a repressor may get in the way of the basal transcription factors or RNA polymerase and making it so they can't bind to the promoter or begin the transcription. And coming to the binding site, Coming to the binding site for transcription factors are often close to a gene promoter. However, they can also be found in other parts of the DNA. Sometimes it is very far away from the promoter and started to affect the transcriptions of the gene. And the flexibility of DNA is what allows transcription factors at distance binding site to do their job. The DNA loop like cooked spaghetti to bring far off binding site and transcription factor close to general transcription factors or mediator proteins. So we can see here in the diagram. So these are the activators and the repressor. These genes will only express if only both the activators are present and the repressor is absent. So here we can see activator present and the repressor is absent, and the transcription will take place. and then here only one activator is present, then only little but little or there will be no transcription will be taken place, and then activator presence and the repressor also will be present. So these are two are the activator and this is the repressor, and then there will be no transcription. So let's talk about the example, that is the modular mouse. As an example, let's consider a gene that is found in the mice called the TBX4. And these genes, what happened, they are very important for the development of many different parts of the mouse body, including the blood vessel and the hind legs. During the development, what happened, the several well-defined enhancers drive the TBX4 expression in different parts of the mouse embryo. And here we can see the diagram. The diagram below shows the sum of the TVX enhancers, TVX enhancer, each level with the body part where it is produces the expression. So what happened? The evaluation of the development, the enhancer like those of the TVX gene, TVX4 gene are called as a tissue specific enhancer, and they will control this tissue specific enhancer like they control a gene expression in a certain part of the body, and the mutations of tissue specific enhancer and a silencer, they actually play a key role in the evaluations of the body form. So here we'll talk about the post-transcriptional label. So here we can see the 5' capping, 3 polyadenylizations, and the splicing will be taken place. And the antisense silencing. The antisense, it is nothing, it is a non-coding DNA strand of a gene. A cell that is uses the antisense DNA strand as a template for the producing the messenger RNA that will direct the synthesis of the protein. The antisense, what happened, it can also refer to a method for the silencing the gene. So here we can see the diagram. These are this red color strand, they are the sense strand, and these are the anti-sense strand. It is like 5' to 3', and this is the antisense from 3' to 5'. So the sense strand has some base sequence as transcript mRNA, except that T is replaced by U. RNA polymerase enzyme will add the RNA nucleotide to sugar phosphate background in the 5' to 5' to 3' direction. Here we can see the mechanisms of the antisense technology. The DNA will form the mRNA and then it will be translated into the protein. But here what happened, this mRNA will form the RNA-RNA hybrid. By the binding of the antisense will come and bind to the RNA and it will form the RNA-RNA hybrid, and then with the help of the enzyme, that is the RNase H, and this H means that this is a hybrid. And then what happened after binding this RNase H will come and break the strand, the RNA-RNA hybrid, and then what happened, it will not translate it, will not the translation will not take place, and then it will not translate into the protein. Let's talk about here the post-translational label. The proteins can undergo a variety of the modifications, such as being chopped out or tagged with the chemical groups. This modification can be regulated and it can be affect the activity or the behavior of the protein. And some example of the modifications are like folding glycosylations, disulfide bond formations, phosphorylation, acetylation. And here we can see in the diagram, the DNA will be transcribed into RNA, and from the RNA will be translated into the amino acid chain, that will be the folding will be taken place, and then it will be translated into the protein. These are the references.